Hello everyone, I'm Hannah and this evening I'll be your host for the Leicester edition of Pint of Science. I'd like to welcome everyone and hope you've all had a wonderful day and are excited to learn more about the beautiful mind. Whether you're from a scientific background or just want to learn more, we hope these talks are something for everyone. We have 3 speakers this evening all talking about different aspects of the beautiful mind, ready to impart their knowledge with us. And if you have any questions or comments, please leave them in the comment section of YouTube, as we have a bit of time after each talk for a few questions, and we hope to engage with as many of you as possible. You can also find Pint of Science on Facebook, Instagram and Twitter. So without further ado, I'd like to introduce our first speaker, Dr. Steven Jacques, who is Head of Anatomy at the University of Leicester. Please take it away, Steve.
Thank you ever so much, Hannah, for that introduction. I'm really pleased to be presenting here at Pint of Science this year. What I want to talk to you about is a very brief introduction, if you like, to the nervous system, but not an introduction about its function as such, but an introduction to how we know about its structure, and by implication, how we know, therefore, about its function. Now, I am a neuroanatomist, which means that I am particularly interested in kind of solving this problem. And that problem is, how do we go from this relatively homogeneous looking blob of matter, which is the brain, and by the way, this is a chimpanzee brain, how do we go from the brain to the complex looking wiring diagrams that we tend to see in popular. So how do we go from this to this? How do we map the brain? And that is a big part of what I do as a neuroanatomist, how we map the connections and the various cell populations within the brain itself. Now, I'm basically going to talk to you, give you a potted history, if you like, of neuroanatomical methods. And then I'm going to talk about some really bang up to date methods, which are really quite exciting and still in their infancy. Traditionally, and probably some of the first methods that we used to study the brain was by dissection, where you take a brain out and you cut it up in a systematic fashion, trying to identify the various bits and pieces. Now, dissection is very nice. You can see the various structures, but you cannot make many inferences about the function of those different parts. In order to do that, what you might want to rely upon is things like natural experiments. This unfortunate chap here has had a stroke, and if he were to die, we could take his brain out and look at the particular area of his brain that has been damaged to cause his particular symptoms. So this could help us to work out what those various bits of his brain do. However, that's a really blunderbuss approach to studying the structure and function of the nervous system. For a much more precise, higher resolution study, if you like, we need to go to animal models. And what we can do in animal models is we can selectively and very precisely either destroy or electrically stimulate a part of an animal's brain, in this case a rat, and then we can look and measure the changes in that animal's behavior in response to that stimulation. And that has given us a huge amount of data and helped us to treat terrible diseases such as Parkinson's disease, for example, by using animal models. As many of you will be aware, there are many beautiful imaging methods as well at our disposal, including diffusion methods that show us where the various axons run within the brain and spinal cord. We've got the functional MRI, which is very trendy, and you're seeing that all over the press lately. These areas that are lit up are essentially areas of the brain which are activated, I think, when someone is watching TV, in this case, that being the visual areas at the back and the auditory or listening areas at the sides. Furthermore, we can use magnetic stimulation to study the the function of different areas of the brain in a living subject. So we can magnetically stimulate a part of the brain and look, for example, for muscle twitches, or we can even stimulate bits of the brain very strongly and switch them off temporarily, which is really quite disconcerting for those subjects. Furthermore, we can, in experimental animals, inject dyes into neurons and see where those neurons run within the nervous system. And what this is showing actually are the cells which supply taste buds in the tongue. So these are, this is a sample of the methods that we have at our disposal to study the nervous system. However, what I thought you would be particularly interested in finding out more about this evening is the science of connectomics. Now, connectomics is something which has only been trendy for about the last decade. And what connectomics strives to do, and this is a very lofty aspiration, is to map every single connection within the nervous system. And the way you can go about doing this is in principle very, very simple. You can take small chunks of brain tissue and you can slice them into very, very thin slices and look at the face of this sliced block down through an electron microscope. And you can then make these beautiful, gorgeous 3D reconstructions of every single neuronal element within that little chunk of tissue. And once you've laboriously gone through that, you can create things like this at the top left side of the screen. This is a small piece of retina, and this is mapping every single neuronal connection within that small chunk of retina. So this offers a very, very high resolution, in fact, the ultimate in resolution, if you like, when it comes to neuroanatomical methodology. So let's think a little bit about the practicalities of this. The full connectome has been established for a tiny little worm called C. elegans. C. elegans, and this is to scale. This is a human brain, a plastic model, and this is C. elegans up here. It's not a bit of dust on your screen. That is the worm. And C. elegans possesses 302 neurons. And scientists have established every single connection between these 302 neurons within the C. elegans nervous system. Now, you might think on the surface, this must be a very trivial matter. However, it took 12 years to establish this map of connectivity. So this map of connectivity, the connectome of the C. elegans nervous system, took 12 years to produce. The human brain possesses around 100 billion neurons. And you don't have to be a mathematical genius to work out how much longer it in theory would take to establish the human connectome based upon the speed it took them to establish that of C. elegans. Of course, technology is constantly progressing, and right as we speak, there are automated machines running, slicing human parts of human and mouse brain, establishing the connectomes of those. But still, it's a gargantuan effort. And I'm coming to the end now of what I'm going to talk about, but what we need to think about is what might be the implications of this. But even before we think of the implications, we need to think, what would we do with this? So imagine if in theory we established the human connectome, and if we fill petabytes and petabytes and petabytes, in fact, I don't even know what the next word is in the orders of magnitude, all of those petabytes of data filled a gazillion hard drives, we wouldn't actually know what to do with it. If you think about the Human Genome Project, we still don't really know what to do with all that data. We're starting to make some sense of it, but we really hardly stand a chance at all of knowing what to do with this connectome data as things stand at the moment. But let's just indulge ourselves a little. Let's think what the implications might be if we established the human connectome. Might it be that people may become immortal? Could it be that if your connectome could be established immediately after you had died physically, could you then be uploaded and your brain continue to run in a simulation in silico? Might we be able to improve upon the connectivity of the human brain? Might we be able to produce super intelligences which may one day surpass our own intelligence? Maybe that would be good. Maybe that would be bad. It would depend upon who you talk to. Might we be able to exploit this? Maybe if we get extremely high resolution imaging data, might we be able to exploit this in order to study mental illnesses? Because what we do know that is that most mental illnesses have absolutely no manifestations when it comes to looking at the physical brain. It is almost certain that mental illnesses will manifest in subtle changes within the connectome. So I do think that connectomics is tremendously exciting, but we've really got to be realistic in terms of what we're going to do with this data. But one day, I think, this will become a reality, and then we're in for some really exciting times. So that's all I've got to say. Thank you ever so much for listening, and I would be absolutely delighted to take any questions that you might have, either right now or at the end. So thank you very much, and that's all I've got to say.
Thank you, Steve. That was really interesting. And then we'll go to the audience. If anyone wants to ask any questions, hopefully they'll flash up on screen.
Okay, Cardi Kamara asks, so hello from Manchester. How do we think that new animal went? Yes, I think that that's a very, very interesting question, Cardi. I think that increasingly we are realizing that there is not a sudden gap between humans and other animals. Okay, so there is no discontinuity between humans and other animals. And most definitely we know, for example, that other great apes are no longer used in animal research, and that is a good thing for sure. But still, it's not been resolved. And still, we're very much working on a kind of utilitarian philosophy here, trying to maximize the greatest good for the greatest number. But I think it's going to have some very, very interesting implications indeed.
Brilliant, thank you. Do we have any more questions?
Tim asks, do I know if memories are stored 1 euro each? That's an excellent question, Tim. The history of memory storage is a very, very interesting field. Back in the 60s, it was in fact believed that memories may be stored in mRNA molecules. And there was a huge amount of research went into looking at the sequences of mRNA, trying to transfer memories from one animal to another. We now know that memories in the sense of, for example, your memory of a fact or your memory of a place cannot be stored in a single neuron. We know that neurons club together in what are called neuronal ensembles, which might be tens to hundreds or even thousands of neurons, which work together to encode a given concept, a given place, a given emotional association. So neurons collaborate together, they form these networks, and that is how we encode memories. As for what is the correlation between the structure of the neuronal ensemble and the structure of the piece of information, that is a very, very difficult problem to solve. It is not the case that the letter X, as you read it, is represented by an X-shaped firework display in your brain. It is far more complex than that. But that's a fascinating question. Thank you.
Brilliant, thank you. I think we have time for a couple more, so if you want another one, here we go.
Okay. What new technologies am I excited about in the future apart from connectomics? Well, Emma, I'm very, very interested in optogenetics as well. Optogenetics is a fascinating technique whereby what we can do is we can use genetic engineering methods. We can use viral gene therapy vectors to insert photoreactive proteins into neurons. So you can imagine we could, for example, identify the molecular characteristics of a set of neurons which are involved in the fear circuit within the brain. We could then design a virus which infects those neurons selectively and which makes those neurons responsive to light. Then we illuminate the animal's brain, and we should, in theory, be able to induce a fear response. And we could then try different drugs which could alleviate that response. So I think optogenetics is one of the most fascinating areas, as well as connectomics. And that has seen an explosion in its use over the past 10 years.
Brilliant. Have we got any more questions, or shall we move on to our next speaker? Oh, we've got another one.
Pinky purple galaxy, fantastic. What can we expect to discover about the brain with our current rate of science in the next 10 to 15 years? Well, that's a very, very interesting question, really. And I think probably the best way I suggest you try to think about that is think about what the world was like 15 years ago. Now, I don't know how old you are, Pinky Purple Galaxy, but 15 years ago, actually, the world was really quite a different place. For example, you almost certainly have a smartphone. And 15 years ago, I remember watching Star Trek and thinking, will we ever have something like one of those things that they're tapping on the screen? Will we ever have anything like that? I think it was a bit longer than 15 years ago. And then, lo and behold, what have we got now? Everybody contains a fully powered computer in their pocket. So that is the way I suggest you think about this. Look at what it was like 15, 20 years ago, and then think, you probably can't even imagine what the world's going to be like in 20 years' time. So I'm sorry if that wasn't particularly helpful, but I think it's about the most realistic answer I can give you.
No, brilliant, thank you. Have we got any more, or shall we? I think we should probably move on. So thank you so much, Steve, for your talk. That was... Oh, no, we've got another one flashed up.
This is great. If you could explore one MH connectome, which I'm not sure what MH stands for. I'm assuming we mean the connectome of an animal, or what, Rachel? Would anyone like to just elicit what MH stands for?
Oh, I don't know. I don't know. Should we move on to another question?
Yes, sorry, Rachel. Mental health. If I could explore one mental health connectome, which one would I choose? Oh, that's a fascinating question. I think I would probably start with the psychotic disorders, actually, Rachel, because we already know quite a lot about the putative structural changes related to those, and it would be likely that we would get the biggest signal if we looked at psychotic disorders and bipolar disorder. And I think also we know that they have probably one of the smallest environmental components. So I'd probably look at schizophrenia or bipolar, Rachel.
Brilliant. Thank you again. We'll move on to our next speaker. Thank you so much, Steve, for answering all of the questions. And to the audience, please keep the questions coming. They've been really, really interesting. So we're moving on to our second speaker now, who's an associate professor in the Department of Molecular and Cellular Biology at the University of Leicester. So I'd now like to welcome Dr. Salvador Macip.
Thank you, Hannah. Thank you, everybody, for coming and listening to this talk. It was an excellent talk, this first one. And we are going to talk now a bit about aging and the brain. So in a way, it's not the same kind of immortality where that Dr. Jacques was talking about, keeping our thoughts in a machine, maybe. Here, what we're trying to do in our lab and in many labs is try to see if there is a way to prevent this age-dependent deteriorating of cognitive functions, these declining cognitive functions that we see in aging. So aging is a process that is now very well studied. So we start our lives looking like this, all rosy and cheeky and nice. And if we're lucky, we're going to end up like this. And that's the process we call aging, this degeneration of all our tissues, this change in our aspect is what we recognize as aging. The process of aging, now we understand it only also not only as a thing that happens to us as a whole, but also with things that happen at the molecular and the genetic level and the cellular level. So we are going now trying to study this process that we see as aging as a series of events that are related to changes in our cells and our genes. That's what we call the process of aging, the biological process of aging. And more importantly, once we understand why this is happening, what we try to do now is try to see if there's any way to stop it or reverse it. And that's how whole this area of research called anti-aging it was born once we started understanding why aging happens. So the goal would be to reverse as much as we can aging or stop it somehow and try to look all like a young, nice Brad Pitt for as long as we can. So what we're talking now is trying to find ways to stop this process at the molecular level, like I was saying. So if aging at the level of the organism is quite noticeable, so we can very easily see how the changes apply to all these different stages in life until we become old. if we can look at our organs, we will be able to see the same thing of changes. So we basically, the answer to the question of why we age is that we age because our organs age. So a liver, a brain, a kidney, when we're young, looks completely different to the same organ when we are old. So this can also be noticeable just by looking at the organs. But if we go even deeper and we look at the structure of these tissues, we also see differences. So our tissues when we're young, when we look at the microscope, the cells look in a certain way. When we're old, the cells look different. So that's the final explanation or the final answer to why we age. We age because our cells age. And that's important because now we have a target. So can we find a way to reverse this aging at the cellular level, so then the organs will be younger and then we will actually look and be younger? So if you look closely at this graph here, you would see that there's something called the senescent cell, these sort of pink cells that show up more and more as we age. So we start with almost no senescent cells. As we age, we're going to get more and more of those. So senescent cells are going to be one of the main features of the rest of the talk, and we can call them the bad guys of this story. Senescent cells are actually have been called the zombie cells for various reasons. First of all, because like zombies, they would not follow orders. They would do whatever they want. And also they would disrupt everything around them. And not only that they are able to, if they bite a cell next door, they may be able to turn that cell into a zombie as well. So senescent cells are bad. Having senescent cells in your tissue is not a good idea. But unfortunately, that's what happens as we age. So we could say that senescence is one of the causes of aging. It's an important factor of aging. And if you look at this representation of a tissue of a mouse, of an animal, when you're young, our tissues look all this nice and organized. As we get older, we would see these senescent cells, these zombie cells just appearing in the tissues more and more and disrupting the function of all the cells around them and even turning the cells around them into more senescent cells. Now, this experiment in 2011 kind of changed the field of aging, because this was the first time that they showed that if you prevent this accumulation of senescent cells as we age, you can sort of revert or stop aging. So this was an experiment, a complex genetic experiment in mice that basically showed that getting rid of senescent cells kept the animal much younger. So then we all started in 2011, we all started to think, how can we apply this to humans? Is there a way that we can find a system that would eliminate or stop this accumulation of senescent cells that we see in aging? Can we get rid of these zombie cells and then remain younger for longer times? And in my lab, in many other labs, we started looking for explanations and targets for this to try to achieve this goal. And we focus on this protein called BTK. It's a long, complex name, and this is a picture of how the protein would look like. But this is not relevant. The important thing of BTK is that BTK is quite involved in the process of senescence. So here you see these pictures. On the left, you would see what is a young cell, how a young group of cells looks like under the microscope. And if you look closer, you would see that when they are old, they look different. They look like longer and flatter, and there's less cells as well, as you can see, because these cells are not dividing. So there is a noticeable change as the cells get older. Now, if we use something that we call a BTK inhibitor, a drug that blocks this protein, you see that the young cells look the same as the other cells, not treated cells. But as these cells age, they don't become old. They don't look the same. So what we basically managed to do here is by blocking BTK, we managed to block senescence. But this is very nice and easy to do in vitro. This is done in a culture plate in the lab. So it's a very artificial system. Our next question was, can this translate into the same thing in an animal model? Can we prevent an animal from accumulating senescent cells and therefore keep that animal younger for a longer time? And this is what we did with a mouse model. So we used this drug called ibrutinib, which is a BTK inhibitor, and we use a control mice that was not treated. And as you can see in this graph, this is a survival of the animals over a certain number of days. You see that the red curve is the animals that are treated. They lived much longer, so they reached to 2030 days, whereas the non-treated animals only reached to around 200. So there is a difference. Obviously, we didn't manage to turn these animals into immortal animals, but we did manage to prolong their lifespan quite a bit. But more importantly, it's not only that we made them live longer, but we made them live better. So if you look at this, this is the frailty score. This is a way to measure how healthy an animal is. Over time, you see that the animals that are not treated, eventually they become frail, they become old, and they are just before dying, they just quickly go into this sort of decay. But the animals that were treated with the drug, with ibrutinib, they remain healthier for longer. This is another way to show this difference. So it looks like this drug managed to keep the animals going for longer and better. So it did delay up to a point aging in these animals. And we wanted then to focus on the brain. So is this, we're actually studying different tissues in these animals. And now we want to see, is this happening in the brain? What are we doing to the brains with the use of this drug? Are we managing to keep these brains younger for longer? And to do that, we used this test called the Barnes maze, which is basically this flat surface that you see in which you have a box in one of the holes. So there's a series of holes, but only what we call the target hole has a box underneath. And the animals don't like to be on a flat surface, the mice, and they always try to find a place to hide. And this is what you see here, the setup in the lab, where you put an animal in the middle of this maze, or this surface, and the animal would try to find a way out. So we teach the animal to find the way out, and the way out is always in the same place. So you see here there are some cues, some marks on the wall, so the animal can recognize that the box is always going to be in the same place. And we teach the animals over a couple of weeks to find the box, and these animals manage to find the box pretty quickly. So what we measure then, after 14 days, after some time, is whether these animals remember where the exit is. And this is a test basically for memory, to see how well these animals remember. And memory is one of the first things that goes in age. So all animals tend to remember less. So what we see here in this test of spatial memory, we'll just put these videos, see if I can turn on these videos. Should be able to. In what would happen here, so these videos don't seem to start, but what you would see here is that this researcher would leave the animal in the middle of this surface, and this animal would try to remember where the exit was. And it could not. It's going to take a while for this animal here, which is an old animal that has not been treated, to find where the exit is. Now, this animal that's been treated with the drug would find the exit right away. And this is how you measure it in seconds. So the average of the non-treated animals would be 20 seconds, around 20 seconds to find the exit. The treated animals would be finding the exit much faster, which means that they remember much faster what was happening, what was going on. So this shows that with the drug, what we're doing is maintaining the memory of these animals, therefore preserving one of the cognitive functions of this animal. And this is something that we also tested with this experiment we call the elevated plus maze. In this case, what we have is this surface. This is like a cross. It's a plus. And these arms here are closed, meaning that they have walls. So the animals can hide in here comfortably. They like hiding in here. And these arms here don't have walls, meaning that the animal can see the rest of the room and could actually fall. So animals don't like to venture into the open arms, so they like to stay in the closed arms. Now, young animals would do that, would explore and get out and more often, because young animals are braver and they would like to explore the world a bit more and then go back to the closed arms. Now, as they get older, they would not do that. They would much prefer to stay in the protected zone. And this is something related to what they call the anxiety-like behavior, which is something that also increases in aging. As the animals get older, they would get more anxious and would not like to explore and be adventurous. Now, if you look at the measurements here, this would be young animals, that how many times the animals would enter the open arms. And these are the control animals and the treated animals, and you can see that more or less is the same. Or how many seconds do we spend exploring in the open arms? But as the animals became older, you would see that the non-treated animals, none of them would like to just go out into the open. All of them would stay in the closed arms. Whereas the treated animals, they would be more adventurous. They would still like to explore. They would still would like to go out. Not as much as when they were young, but still they would like to see the world a bit more than the untreated animals. So what we're showing here is that we manage to preserve another cognitive function with this drug. So this drug, what it's doing is reducing this anxiety-like behavior that's associated with aging. So we have both memory preservation and this sort of anxiety, preventing this anxiety associated with aging. These are two cognitive functions that we manage to reduce or preserve with this BTK inhibitor called ibrutinib. Now, so what we showed here is that there are ways to stop the biological, or delay at least, the biological process of aging in the brain. Now, the question is, can we do this in humans? Is there a way to reduce or prevent this degradation of the brain, or this loss of cognitive functions that we see as we age? Can we just turn this into a much slower rate of decay by using drugs, by using ways, for instance, to prevent the accumulation of senescent cells, stopping senescent cells from happening? Obviously, this works in animals. In humans, is a bit more complex. We would have to use drugs that would have no side effects, so or at least not important side effects. So this is something that we're going to be exploring. And what we do in our lab, it's similar to what they're doing in other labs with other drugs. So my guess is that in the future, in the sort of short to mid-term future, we will see drugs that manage to stop the biological process of aging, and therefore revert or delay some of the this degeneration that we see in tissues, whether it's the brain, it's the liver, the muscle, or whatever. But we are close to a drug that could be tested in humans in the next years. And obviously, this by no means would mean that we would stop aging or that we become immortal, but we may be able to live longer in better health. Like we saw with these animals, the point is not to live longer, but to live better. So the years that we spend in old age, spend them as healthy as we can. And drugs like this in the future should be able to help us. So the work I showed you today has been done by this wonderful team that we have in the lab. Most experiments were done by these clever PhD students that we had in the past in the lab, and now this project is being taken forward, but some other PhD students, Amal, Han, and Fares, and we're trying to see whether we can understand better why we age, why these animals age, and how we can stop this aging, and whether that can be turned into something that could be applied to humans. And I'll be very happy to take any questions you have now. Thank you very much for listening.
Thank you, Sal. So any of the audience want to ask any questions, please go ahead. Wow. So, wow, all the way from India. Hello. If our cells renew themselves on a regular basis, then how is it that the new ones replacing the old ones are supporting the process of aging?
That's a very good question, Rama. And actually, that would be the plan. Ideally, all our cells should be able to be replaced and renewed when they become old. And that does happen when we're young, and our tissues remain sort of fresh and healthy for quite a while. But the human body has not been thought to live more than 30, 40 years. That's sort of our normal average lifespan in the wild. The problem is that when we get into old age, all these processes that work very well in the first stages, in the first decades of life, stop working. And that's where this accumulation of senescent cells happen. During the early years of life, the senescent cells, if they appear, they are cleared and replaced by young, fresh cells. As we age, this doesn't happen. Why? We don't know. And if we can find out why and find a way to keep the tissues always regenerating, in theory, we could become immortal. That's actually what happens in certain animals, like the jellyfish, that manage to keep replacing all these cells as they become old. But in humans, that is not possible. But definitely, we're trying to understand why this is the case and why this happens. And perhaps in the future, we should be able to at least try to keep to regenerate our tissues for longer.
Brilliant, thank you. I think we have enough time for one more question, if that's possible. So is there a tipping point when it's too late to regenerate?
That's a very good question as well. And it looks like, well, it's difficult to say exactly when aging starts. So probably aging starts from the moment you're born. But there has to be probably a point after which, no matter what you do, there's no way you can get rid of, well, even if you get rid of these old cells, nothing's going to happen. We are testing that in our mice now, trying to see whether starting the treatment at different points would be useful or not, and whether there is a point of no return, as you may say, that after which there's no way that whatever you do, the tissue is going to be able to regenerate.
Brilliant. Thank you, Salvador. That was a really good talk. And we'll now move on to our last speaker of the evening, who is a lecturer in the Department of Neuroscience, Psychology and Behaviour at the University of Leicester. And please, when you're ready, Dr. Eoin McElroy.
Talks so far. My talk's going to be a little bit different. I'm a psychologist. And I'm interested in how we think about and how we measure things like mental health problems and diagnosis. So I'm going to spend about 10 minutes talking about the history of psychiatric diagnosis and how scientifically valid and useful they are, and discuss some newer evidence-based alternatives. I think it's fair to say that mental health difficulties affect us all. Chances are we've either experienced mental health difficulties ourselves at some point in our lives, or we've witnessed someone close to us suffer from these problems. And when we talk about mental health problems, we usually use terms like depression, anxiety, schizophrenia, PTSD. Psychiatric diagnoses such as these are part and parcel of everyday conversation. But what are psychiatric diagnoses, and where do they come from? What do they look like? Well, here's an example of the diagnostic criteria for major depression. Most diagnoses, they are assigned by clinicians, and they're based on quite detailed symptom checklists from diagnostic manuals, such as the DSM-5 and the ICD-11. So to qualify for a diagnosis, people typically need to display a set of core symptoms for a defined period of time, weeks or months, along with additional symptoms. And these symptoms must result in clinically relevant distressed or impaired functionality, so impacting how you live your day-to-day life. So in essence, a diagnosis or diagnosis in general is a language. Terms such as depression, panic disorder, they quickly sum up a series of experiences and behaviors that typically co-occur within an individual. And these terms they're proposed to reflect an underlying illness or disorder. Why do we do this? Well, there are a number of quite pragmatic reasons for this. For instance, by developing a common set of descriptions for specific mental health problems, we improve our consistency and comparability. This makes it possible to compare trends in mental health problems across different groups and time. Think of, across different countries, what are the levels of depression in different parts of the world. And by having an accurate description of a quote-unquote illness, we are better placed to understand the factors that cause that illness. And also by knowing what specific disorders look like, we can formulate treatment strategies that are tailored to those specific conditions. So if someone presents with diagnosis X, we give them treatment Y. And this should help people get the most effective treatments for their condition, which should increase their likelihood that they make a full recovery. And now we come to the crux of the matter. Are these categories any good? Does the scientific evidence support the idea that these unique psychiatric disorders exist? So let's revisit some of those aims I just discussed. So the rationale is to have clear-cut, consistent categories so we can describe and compare trends in mental health problems. Well, one of the biggest problems in this regard is that people very rarely fit into one diagnostic category. Comorbidity, meaning basically meeting the criteria for more than one disorder at the same time. This is the rule rather than the exception. And studies have shown that basically if you meet the diagnostic criteria for one disorder, you have a 50% chance of also qualifying for a second disorder. And if you have two disorders, you have a 50% chance of meeting the criteria for a third disorder. And what this does is it suggests that the boundaries between our specific diagnosis are not very clear-cut. And it's not just individuals who have problems fitting neatly into our diagnostic categories. Clinicians do as well. What we call inter-rater reliability is quite for diagnosis is actually quite low between clinicians. So in other words, studies suggest that if you take two psychiatrists and have them diagnose or assign a diagnosis to a patient independent of one another, roughly 50% of the time they're likely to assign an entirely different diagnosis or two completely different diagnoses. And what about treatments? Again, I talked about how the whole point of psychiatric disorders or diagnosis is to kind of, develop very specific treatment plans. But what we see in practice is a remarkable lack of specificity. So, for instance, common treatments such as, say, antidepressants, these have been shown to be effective, as effective for treating things like anxieties, phobias, as effective as they are treating depression itself, more so in some cases. Common therapies, talking therapies like CBT, cognitive behavioral therapy, which is very commonly used. This can be used to treat anything from depression, anxiety, all the way up to kind of psychotic disorders like schizophrenia. So again, it doesn't look like our diagnoses really tell us anything about how to treat people. Okay, but are we getting closer to the root cause of mental health problems by looking at these diagnoses? And again, we're seeing remarkably little progress in this area. So genetic studies, for instance, have consistently found limited evidence that genes can lead to specific disorders. Any genetic effects we tend to find, it's usually many different genes predicting many different types of behaviors. Like Steve talked about earlier on, there are no real biological tests for specific mental health problems. You can't scan somebody's brain and say, okay, this person obviously has depression, this person has schizophrenia, etc. Other risk factors, things like traumatic life events. So this is an area I do some research in. Again, we see a lack of specificity here. Having a very traumatic event in your life, like say physical abuse or sexual abuse, this can lead to a range of mental health problems. It's not the case that one specific type of trauma leads to a very specific type of disorder. So based on the evidence available, it looks like our most widely used psychiatric diagnoses achieve few of their intended aims. So why might this be the case? Well, this brings us back to the title of the talk, Psychiatric Diagnosis, Useful Tool or Bad Science. In many ways, this title is misleading as it implies that some science went into the development of these categories. So starting with the third edition of the DSM, the world's leading diagnostic manual, psychiatric categories were essentially voted into existence based on the consensus of expert panels. In other words, these categories, they weren't informed by scientific data, rather they were largely based on clinical experience and opinion. So why might this be problematic? You might think, well, these diagnoses are based on clinical experience. How wrong can they really be? Well, from a research perspective, if these categories are not precise is the key word here, or if they describe only a small part of a larger phenomenon, can we really think of them as valid, reliable, or in any way useful, I guess? So is it surprising really that genetic or other studies have failed to uncover the origins of mental health problems if we don't know how to properly describe these things or measure them? So I'll give you an example or a metaphor. If we're busy trying to look for the kind of the source or busy studying the kind of the tip of an iceberg, chances are we're going to miss important information that's lurking beneath. So what can we do to improve this? More and more alternatives to classic diagnosis are being proposed, and I'm going to very quickly highlight one approach that's increasingly popular. This is the hierarchical taxonomy of psychopathology, or HiTOP model. And what this is a data-based alternative to traditional diagnosis. What this approach does, it uses data from surveys of thousands of participants, where people are asked to rate the types of symptoms and experiences they have. HiTOP then applies advanced statistical techniques to identify groups of problems that are likely to co-occur. So instead of hundreds of specific diagnoses, what we have are these a smaller number of broad dimensions. So things like emotional problems, behavioral problems, and thought disorder. And these dimensions, they're proposed to be continuous in nature rather than strict categories. In other words, they range from healthy, happy, non-distressing experiences to more clinically relevant distress and impaired functionality. And we can all move up and down these continua depending on various experiences in our lives. What are the scientific benefits to this approach? Well, the empirical structure of this model has been supported by a wide number of studies, over 500. 28 studies have found that high top is useful for things like selecting the right treatments for people. 20 studies have shown that high top improves the reliability of clinical diagnosis. So, clinicians using this approach are more likely to come to the same conclusion about an individual. Compared with traditional diagnosis, this dimensional approach is twice as effective at predicting various outcomes of mental health problems, things like who's most likely to recover and how. And in terms of genetics, looking at these broader dimensions is kind of a more reliable and valid way of doing things, lines up more neatly with genetic studies. And finally, statistical ranges can be developed. So we can develop cutoffs for, clinical cutoffs for people who might need treatment versus people who don't need treatment. And these are backed up by hard science. They're not backed up by arbitrary symptom counts and things like that. So really, it's about introducing a more scientific data supported approach into psychiatric diagnosis. So that is it from me. Hopefully that was interesting. And yeah, thanks for coming along.
Thank you, Eoin. That was really interesting. Does anyone have any questions? So I gather that microbiome has an effect on mood, with poor diversity microbiome leading to depression. What do you think?
Okay, the microbiome, not really my area of research. I guess with traditional diagnostic approaches, one of the points I was trying to make there is that they're developed from this kind of disease mentality from I guess clinical medicine, and the philosophy is that there has to be some sort of root cause. There has to be some factor that causes these mental health problems in individuals. But with a more dimensional approach, like I talked about, we're kind of more agnostic about causality. So we know that mental health problems are very, very complicated things. There's usually multiple risk factors feeding into these problems that cause us to have these symptoms. So for something like poor mood, yes, the microbiome is absolutely something that could feed into this. And yeah, there's a lot of research in this area developing at the moment. But again, does it necessarily line up with something as specific as major depressive disorder? I would think probably not. Whereas maybe a more broader emotional disorder spectrum, we might find something like that. The microbiome has a large role to play in that. Hopefully I've somewhat answered that question.
Brilliant. I think we have enough time for another question. If anyone has one. So is depression passed down through families?
Again, like I said, any sort of mental health problem, you need to think of it almost like a recipe or a complex equation. There's lots of things that feed into it. Yes, there can be some heritability. So in other words, if you are if you have a parent maybe who suffered from depression, you're at increased risk of suffering depression yourself. But this, again, where the causality lies, we can't be certain, and it might not be the case for it's the same for every individual, because we know things that, okay, if you were growing up in a household where maybe there were mental health problems, well, that might have a knock-on effect on your family environment and things like that. So it may be genetics, it may be kind of the environment you grew up in the family, or it might very well be a combination of both. So unfortunately, there's no easy answers for a lot of mental health problems.
Brilliant. Okay, next question. So what are the main reasons for differing diagnosis from one specialist to another?
Again, it's human error. Like I said, if you're dealing with these very, very specific categories, clinicians are asked to rate things like, well, is this patient, I'm not a fan of that term, but is this patient suffering from clinically relevant distress? Or, you know, are they suffering? Now there's an element of subjectivity to that. So obviously this is going to differ between clinicians. So really kind of dimensional quantitative approaches. It's about developing maybe new questionnaires and new methods of assessing symptoms that are data based and can offer us maybe more consistent ratings between clinicians when they're assigning diagnosis.
Brilliant. I think we have enough time for one more question if we have them. So some say that we need to move away from a distress-based diagnostic system. Do you have any thoughts on this?
Well, yeah, I mean, distress. Again, I think distress is always going to be an element of mental health problems. You know, it's what actually constitutes a mental health problem is probably another entire different talk. But distress has to be an element of it, because if somebody isn't feeling distressed, then really can we say that this is a problem that needs to be solved? But I think what we are moving away from is this idea that mental health problems, psychological distress, that these are illnesses, you know, that these are brain disorders that are you know, caused by abnormalities in the brain. I mean, Steve talked about this earlier on. If we scan people's brains, we can't differentiate between a healthy person and somebody who has depression. So I think it's about moving away from this disease mentality and focusing more on people's situations and the struggles they've faced, and maybe inequalities they've faced. You know, have they had a difficult life? Have there been systemic issues that have made things unnecessarily difficult and unfair for them?
Brilliant, thank you. I think we're going to bring everyone back in and ask if anyone wants to give any more questions to the panel. So if anyone has any more questions for everyone. No?
Oh, they take time for people to type, though, don't they? I think people often, you know, I think it's easy to forget that how immediate we used to people communicating with us face to face, isn't it?
Still getting used to the online live. Yeah.
So 2 sticks, that's an interesting one. Mild sepsis appears mental illness. I mean, can I take that one? I mean, I'm happy to take that one. So 2 sticks, you're asking about the relationship between sepsis and mental illness. The clinical term for this is delirium. So we know that in septic patients, due to their infection and the associated inflammatory response, a huge amount of chemical mediators are released, which can interfere with brain function. And as a clinical doctor, I've dealt with a lot of delirious patients over the years, and they can be very difficult to deal with. But the amazing thing is, if you treat their infection, they go completely back to normal. For example, I've been attacked with drip stands and fire extinguishers, and then I put some antibiotics into the patient, and they recover. So absolutely. And this is a constant thing we have to think about as clinicians, thinking about, you know, is this delirium? Is this dementia? Is this delirium overlying dementia? Or is it something else? And this is where your judgment as a clinician comes into it. And I know Eoin is a big advocate of objective measures and that, but of course, the gut feeling of a doctor seeing a patient is very, very valuable too.
Brilliant, thank you. Another question. So do you think having the human connectome could be used to make the ultimate neural network for AI?
Yeah, Vishnu, I answered this in the chat. Essentially, this is a concern of a number of workers. Primarily, I must say, philosophers as opposed to scientists, although, of course, we know Stephen Hawking did have some anxieties about this. And I recommend that you read a book called Superintelligence by Nick Bostrom, who is a philosopher at Oxford in the Future of Humanity Institute. And he worries just about that. If we uploaded a connectome, if we tweaked it so it became super intelligent, could it actually outsmart us? And it might seem like a Black Mirror sort of sci-fi scenario, but we've got to be aware that, as I said before, think about when I was a kid watching Star Trek The Next Generation, I couldn't even imagine something like an iPad. I couldn't even imagine it. And we've got them now in our pockets. Well, actually, not an iPad, an iPhone. But so yeah, you know, the rate of progress is currently exponential, isn't it? So we have to have these philosophical discussions right now. We have to be prepared for this because it could have a nasty habit of creeping up on us.
I agree with you. If I can jump in with the whole ethical implications of the research we're doing. Also, when we talk about aging and anti-aging and stop aging, there's a lot of ethical implications that we should start talking about now. Because as I said, now we're doing this in animals, but pretty soon we should be able to do this in humans. And I'm thinking 10, 20, 30 years, this could be happening. And we need to discuss this before we actually are doing it, because there are a lot of things that could go wrong and a lot of things that we need to understand before we actually do it.
Yeah. And there is a little bit of kind of dismissiveness amongst some scientists to philosophers and the like, but we need to be working together in an interdisciplinary way, because it's not what we can do, it's what we should do which counts.
Brilliant, thank you. Next question. So what can we expect to discover about the brain in the next 10 to 15 years?
I did answer that one actually from Pinky Purple Galaxy. So yes, I think we can move on to a different question there because I did answer that one.
Okie dokie, we'll move on to the next one then.
There are some new ones coming in at the bottom.
Yeah, there are. There's loads coming in. We're very lucky.
Because these are all questions that we already answered. Some of them in the chat.
I don't think that one's necessarily a question. Have we got another one? I think that's one for you, Steve.
Well, for me, can metabolic brain damage be caused from sepsis? I mean, yes. I mean, once again, you know, sepsis is a multi-system disorder. Sepsis can affect all organ systems, and it could, for example, lead to a period of ischemia in the brain, which could affect brain function permanently, or even if the infection itself were in the brain, that could affect brain function permanently. This could in turn lead to psychiatric disorders such as depression. For example, we know that strokes increase the risk of depression in patients completely independently of any effect, any negative effect that having a stroke obviously has on your quality of life. So it most definitely is not beyond the realms of possibility, and it might well not show on any imaging modality. So yes, I agree with you, Two Sticks.
Brilliant. Thank you for answering all of those questions. I think we're going to have to wrap it up for this evening now. So thank you for everyone for participating and speaking, and thank you to the audience for turning up and asking such brilliant questions. So thank you, everyone, and good night.
Thank you. It was a pleasure.
Thank you.
Goodbye.